Hi. Did I really just see someone say third wall with a hairy chest? I thought I did. How are you guys doing? You alright? Ooh, don't have myself on mute. How's everyone doing? Did you guys have a nice, well, I say, did you have a nice long weekend? But I realised that you guys probably spent it doing the exact same thing you normally do. So, yeah, let's all be happy we're alive. I like that sugar beast. Did anyone do anything remotely nice over the weekend, or was it all fairly standard, fairly boring? Watching pages from CFAX. Meme Maker, is that even a thing? Can you even watch CFAX anymore? I swear you can't. And also, I don't know. I'm surprised you know what CFAX is. I feel like that's something even my generation barely had. That, I'm going to Google it. Is CFAX even still going? No. Right, okay, cool. I didn't think it was. But I'm glad you know what that is. It's nice. Nice. Ah. Anyone done anything? Come on, someone's done something. You're a student doing Cambridge International is Snap provides for you. Ah, excellent. Right, someone has asked me the first standard question of the day. So in terms of specifications, I cover the like British ones first. So I'll do AQA, OCR, EdXLA, EdXLB. However, one of my students who is currently like doing the proper Snap Revise on our website. She is a Cambridge International person, and yeah, I've been making Cambridge International fit. It's basically the same stuff, if I'm totally honest. Someone just did a little workout. I am pleased that you're doing little workouts. That is good. I'm glad that you guys managed to find some time to exercise. Revision, classic, true 70s, he knows CFAX. Me? Did you say I'm 70s? Hold on. Yes, this is a highlight. That's nice. Good. Cool. Yeah, I'll start showing my screen in a second. I feel like whenever I do these like sessions, I always start with just my screen and I never actually have just me. So I thought I'd actually have just me for a bit, and then as soon as I change my mind, that's a terrible idea, I'll change it. Well, let's make lunch. Fasting gang. Yeah, one of my friends works for the NHS, or she's a doctor, and she's currently fasting and struggling. And apparently loads of people keeps like buying things for the hospital, so they keep sending like donuts in or whatever. But like a massive population of the doctors are just fasting, so they're really struggling. Ooh, second main question. How long is this? So this is an hour. So I'll be going for about an hour, maybe 45 minutes, around that time. That'll be it. What else? Should you buy the Snap Revise package? Obviously, you should buy the Snap Revise package, but I'll wait to see how you feel. See how you feel after this first. If you feel like you want to do that, then check this out first. Will you carry on doing this after lockdown ends? I don't know. Kang, I am obviously going to say yes. Are we going to do this after lockdown ends? I don't know right now. I guess it depends. Maybe. We'll see. I don't think we've decided just yet. Yes, those of you that have noticed that I'm wearing a hat, you are quite right. I am indeed wearing a hat. My hair, hang on, jealous. My hair is now getting to the length I can't get a haircut, where it's basically going down to my nose. So the hat is there to sort of hide the shame of having my long hair, which I can't do anything about. I decided, this is a good top tip before we get started. Top tip, if you ever go to a festival, if you're ever allowed outside again, or if you ever do like, I don't know, I did the ink trail a few years ago. If you ever do something like that and you can't feasibly wash your hair, hats hide your shame. They're the best things ever. Right, I'll share my screen with you guys. Okay, should be able to see that. I think I can still see you guys. There you go. You love the Beatles. I'm glad you love the Beatles. Wolf can't afford the package. Well, I think I'm going to give you a £10 offer thing. So hopefully that helps. I'm not talking about Boris's speech. I feel like YouTube is still kicking people for doing stupid things. Well, not stupid things, but for having any false information about what's going down at the moment. YouTube is just getting rid of people. So I'm not going to talk about that. Anyway, I know there's no shame in long hair. I agree. It's just annoying me. But I don't know. When I go for a run, I'm just blind and I want to get a haircut, but I can't. So anyway. Right, guys. Weird question, am I a leftist or a rightist? That doesn't feel very relevant right now. Why does it matter? So right, here are some things then just to mention before the end of today. So last week we gave away another free account for Snap Provides. So just so you are aware of how much money you are willing or you're able to basically get off of us for free, you'll basically get a package for Snap Provides, like an ultimate account for, well, until September, basically. So if you are interested in having about £200 worth of education, follow us on share a pic of us on your story of, like, I don't know, you doing something remotely creative. Someone last week used a ruler to produce a phagocyte, and I thought that was really funny. And someone else did something weird with a nose, and I don't know what that was. But yeah, do something interesting, and I'll give one of you a free account. I feel like that's a nice little thing from me to you. Someone who got one of the first ones is now turning up to about at least 2 or 3 lessons every day. So she's getting 2 or 3 hours of education for free every day, which is pretty good. Also, for those of you interested, I've got a coupon code for you at the end of this session too. Only last month. I don't know what you mean. Oh no, I'm not going for the man bun. Someone told me that. One of my friends said that I'm getting dangerously close to man bun territory. And I don't know if I have the self-restraint to not do it. No, it doesn't only last a month. No, it lasts until September. Lasts until September. Or so I've been told anyway. Anyway, right, let's do some questions. So as I said, today we're doing exchange of mammals and plants. So ventilation involves various parts of the mammalian respiratory system. Which of the following statements, A to D, describes inhalation? So rib cage moves up and outwards, external intercostals relax, diaphragm relaxes, rib cage moves downwards and inwards, external intercostals relax, diaphragm relaxes. You can read the rest. What do you reckon? Kang and Toby going bold, Anissa going bold. Loss of C's. Rainier's lost the plot. What is inhalation? Inhalation is breathing, like the inspiration or inhalation is like that. That is inhalation. So you should be able to feel at least some of these. If you're not sure about the rib cage moving upwards, downwards, upwards, or wherever, you should be able to feel. If you breathe in, feel what happens to your ribs. Those of you saying it, you are right to say C. Ooh. So the rib cage moves upwards and outwards. Yes, when I breathe, I can physically feel my ribs moving out and I can physically feel them moving up. If I want to make my ribs move, I need muscles to do that. The ones which you use are the external intercostal. So you essentially have some on the outside of your ribs and some on the inside of your ribs, external or internal. External ones pull your muscles, they pull your ribs out, and your diaphragm needs to contract. So when your diaphragm is relaxed, it is sort of curved like this. That's relaxed. When it is going to contract, it flattens and it basically pulls everything down. So yeah, C is correct. Out of interest, what does this cause? All of these things, what does it cause? Causes air to flow in. Why? Good. Yeah, this increases the volume. So the top half of your body, basically from your abs going upwards, is called your thorax. So the thoracic volume, which is your, as I just said, your abs upwards, your thoracic volume is going to increase massively by doing all of these things, right? If you increase the volume, then the internal gas pressure, or it is gas pressure inside, will decrease. So decrease in thoracic pressure. Nice. Did it? Oh, on PowerPoint I've sent you. Yes, there is a mistake on the PowerPoint I sent you. Apologies for that. You can change it, I'm sure, or you can just ignore it. Figure 1.1 is a diagram that represents inspiration and expiration in a human. So you've got the trachea, ribs, lungs, diaphragm, spine labeled up. Which of the two diagrams, A or B, represents the body immediately after expiration? Describe how this diagram justifies your choice. So which one shows immediately after expiration? So air is moving out, yes. So expiration is air moving out, I agree. So what's got to happen for you to be getting rid of the air in your body? You are right, it is indeed A. Can you describe why it's A? So describe how this is justifying your answer. No, it's not because you breathe in after. That's not got anything to do with it. You can't breathe in until your lungs look more like the one in B. So why does A show you that this is immediately after expiration? Good. The diaphragm, as opposed to being flat, has curved. So here it is flat, here it is curved. So curved diaphragm is fine. What else? Or you could say relaxed or something. So different specs say different things here. So I'm used to saying relaxed, so I'll probably say relax more, but curved is also fine. Decreased lung volume. I guess, yeah, the lungs look a lot smaller, don't they? So yeah, lung volume decreased. Definitely. Ribs are sort of, yeah, look, you can definitely see a difference, can't you? Look at these ribs down here and then look at these ribs down here. Look at the sort of angles that the ribs are going at. So the ribs are definitely facing downwards on the left. They're definitely up and they're definitely out. So the ribs are definitely moving further forward and they're definitely less of a slope in B. So the lung volume has decreased. We could say that lungs are down, sorry, lungs, we could say the ribs. Ribs are down, something like that. That sounds quite good. So what else do I have? Yeah, that's pretty much it. So in my mark scheme, A, because the diaphragm is flat or is relaxed, the ribs are in, the lungs are, they have a lower volume, anything like that, absolutely fine. Okay, so this again is something that some of you will have seen more of than others. So the following spirometer trace shows the results of an experiment. Soda lime was used to extract the carbon dioxide from exhaled air. What is the rate of oxygen consumption in this experiment? How could I work this out? Does this specific stage have a name? It doesn't really have a name. Expiration and exhalation are the same. Oh yeah, I didn't even notice that the units here are wrong. That is a very good point. Well done for noticing that. Yeah, so decimeters cubed. If it's a rate, it can't just be a volume, so it's not that one, right? This also isn't. It is a rate, I guess, but the rate of oxygen consumption is not 12 breaths. So it's not that one either. So it's definitely between B or C. Can you change units? You can't change breaths. Oh, maybe you could change breaths. If you did breaths feasibly, you'd have to, I don't know, 1, 2, 3, 4, 5, 6. I can see why you'd say 12, but we don't know what the volume is. So they want the rate of oxygen consumption, and that doesn't really work. How do you work it out? Those of you that are working it out, how do you work this out? Tangent and find the gradient. How could I do a tangent to this? It's literally changing all the time. Like the tangent there would be like that. Tangent there would be there. Tangent there would be like that. Tangent there would be that. Then that. Then that. Couldn't do a tangent. That'd be crazy. Yeah. So with this, you want to sort of look at, in terms of working out how much oxygen is being used, we're assuming here that all of the carbon dioxide is being absorbed by soda lime. Nitrogen is going to stay the same. The only thing that's changing in the volume of air is going to be what? What's the only thing that's changing in the volume of air over time? If carbon dioxide is being absorbed and nitrogen is going to remain pretty much the same, what's the only thing that's changing? Yeah, the only thing that's changing is the amount of oxygen in the spirometer. So if I start off with, you want to go from either the top of a peak to the top of the bottom peak, or you want to go from the bottom of the trough to the bottom of the last trough. So let's go for the bottom of the trough, right? So over here, it has a volume of 2 decimeters cubed. Right, by my last trough, I am down here, and I'm 1, 2, 3, 4, 5 up. There are 10 little squares, so that's 0.5. Right. Although saying that, maybe I should be doing it. No, that sounds about right. Yeah. So, and the amount of time here, because otherwise I want it to be 30 seconds. Do I have the same amount of time between these 2 points? So I've gone past 0 by 1, 2, 3, 4, 5 little units. 1, 2, 3, 4, 5 little units. So that's fine. So I've gone from 2 decimeters down to 0.5 decimeters, and that has taken 30 seconds. So that is a difference of 1.5. So that means if it's moved down, the only thing that's changed is oxygen. So that's 1.5 decimeters cubed in 30 seconds. I want it per minute. So if I know it's 1.5 per 30 seconds, then I know that it's got to be 3 decimeters per minute. So it's got to be that. For any of you who've never seen this little -1 thing before, it's really important that you realize that that little -1, it just means divided by. So you're probably very used to people using miles per hour or something like that. If you were a scientist writing that, you wouldn't write mph. You might see it written as m-h, miles per hour. That is one way of doing it. Or the best way of doing it would be miles per hour, like that, which you'd never recognize. I reckon if you saw that on like your car, you'd be confused by that. But that's the same as mph. M hour -1 is the same as m-h or mph or whatever. Right, just so you know. Okay, so mammals use lungs for gas exchange. The following passage describes how gases are moved in and out of the lungs. Complete the passage using the most appropriate words or phrases. When air enters the trachea, mucus secreted by. Ooh, well done. Goblet cells. So mucus secreted by goblet cells traps dust and microorganisms. Air diffuses through the bronchi and the bronchioles. Smooth muscle in the bronchioles relaxes during the fight or flight response. Oh, this is getting a bit weird. This response is produced by the sympathetic nervous system, which contains neurons that secrete the neurotransmitter. If any of you know this, I'll be impressed, because it's more of a year 13 thing. Namrata, you are very, very, very close here. So the sympathetic nervous system, this is the one which is going to speed up your body. So you're very close to, say, adrenaline, but the neurotransmitter, which is very similar to adrenaline, has a weird little word in front of it. Good guess with GABA, good guess with acetylcholine. You sympathize with that. Nice game. Noradrenaline, Olivia. Thank you. Okay, right. Ignoring that question, because it's a bit of a weird one. Basically, if you can know it for the future, right? Your nervous system, the thing that speeds up your heart and the thing involved in fight or flight, the neurotransmitter is called noradrenaline. It acts like adrenaline. If you are American, which I don't know if any of you are American, and if you ever go to university and do biology or medicine or veterinary or anything like that, you'll see it not written as noradrenaline, but you'll see it as norepinephrine or epinephrine for adrenaline. It means the same thing. Exactly. Right. During inspiration, both the something and external intercostal muscles contract. Good. The internal intercostal muscles only contract when expiration is. Forced. Lovely. Okay, so this is something which is a bit different to what we said earlier then. So when you breathe in, your external intercostals need to contract, right? To breathe in, your external intercostals need to contract. When you breathe out, most of the time, you don't actually need to contract your internal intercostals. So when you breathe out, most of the time, it's just the elastic tissue in your alveoli recoiling, and that is enough to move air out. Like some of you, do any of you ever go to like a science open morning, or have you ever done one at school where someone has like a big old set of lungs on a table and you can blow air into them? Have you guys ever seen that before? No? Oh, right. In which case, Phoebe Carter, are you one of my old students? There is something you might have seen at some point in your life, and you can go on YouTube if not, where basically you blow a whole load of air into some lungs. No. Oh, okay. I used to teach someone called Phoebe Carter, which is weird. You breathe some air into some lungs, and basically the lungs will expel all that air out again. They don't need to have any muscles to do it, right? So the only time when expiration is using the intercostal muscles is when it's forced. So when you're properly going like, that's the only time when you're going to use your muscles to do it. Oh, sorry about that, Phoebe. Okay, cool. So the lungs in a mammal are adapted for efficient gas exchange. The diagram below illustrates a small part of the lungs responsible for gas exchange. So these are your alveoli, as I'm sure you know. On the diagram, add a line labeled P to a branch of the pulmonary vein. Yes, Sard, I used to do the same thing. I used to get little kids, little year 6 and year 5 kids at open evenings, so I'd blow into it. There's always one kid that sucked because they never realized it, blowing and sucking different things. So I always like sucked up a bit of lung juice, which was pretty gross. Is this one of the exceptions? It could be, could be. So which one is showing you the pulmonary vein? Yes, so you guys are right. Right, so essentially what you've got, as I'm sure lots of you know, is you have the heart, you've got the lungs, which are going to be, let's put them up here on my diagram, and essentially blood is going to be leaving from the right-hand side of your heart through the pulmonary artery, goes through a pulmonary artery, and then it goes back into the aorta of the left side, so this is the pulmonary vein. Cool, well done. The diagram below represents the approximate concentrations of oxygen and carbon dioxide inside an alveolus and a capillary in the lungs. These gases will diffuse at different rates. Use the information in the diagram, explain the difference in the rate of diffusion of these two gases. So which one's going to go faster, which one's going to go slower? The hepatic portal vein is another weird little exception to that rule as well. Anyway, so oxygen is faster. Why is oxygen going to be faster? Good. Well done, guys. This is impressive. So using the information, explain the difference in the rate of diffusion of these gases, right? Let's get the mark scheme up so I can definitely make sure I'm telling you exactly what it says. Okay, cool. So you need to say which one's faster than which first. So first mark, you want to say that the rate of oxygen is faster than the rate of CO2. This is a really nice, easy question. So be pedantic with this. If you have an exam question, don't just say the rate of one is faster or conversely the rate of the other one is slower. Try and mention both because examiners love to take marks off you for things like this. So the reason why then, as you guys have just said, is because there is a steeper concentration gradient with oxygen than there is for carbon dioxide. Let's go for concentration gradient. So yeah, the difference between carbon dioxide is like 2. The difference between oxygen and, well, the difference of oxygen is significantly more, so it's 7. Doesn't tell you partial pressure. It doesn't tell you the partial pressure, no, but we are assuming that because there is a large amount of oxygen here and a fairly low amount here, the rate is going to be greater because there is a greater concentration gradient. So you guys should all, well, not all of you should be aware of this, but some of you will be aware because it's in some specs. The rate of diffusion is proportional to surface area to volume ratio multiplied by concentration gradient divided by diffusion distance, right? That's Fick's law. Essentially, if you were to double the surface area to volume ratio, you're going to double the rate. If the concentration gradient is twice the size, which this basically is, then that's going to make the rate about twice as quick. If you were to make the diffusion distance greater, though, you'd actually reduce the rate. So if you double the diffusion distance, you halve the rate. Feasibly, they could have got you to actually work that out. They could have got you, for those of you that do Fick's law, you could feasibly have worked out that the I don't know, the concentration gradient is however many times greater with oxygen than it is with carbon dioxide. Okay, so that's about it. So is PO2 and oxygen tension the same thing? Partial pressure of oxygen is basically the concentration of oxygen. So essentially what's going on is because a concentration of oxygen is quite hard to calculate, you can just instead sort of change it for the pressure produced by oxygen. So the more oxygen there is, the more pressure there is. So partial pressure is kind of used, someone will probably correct me with this at some point, but it's kind of used interchangeably with concentration because there is this proportional relationship between concentration and partial pressure of oxygen. So as one increases, the other will increase. Steep concentration gradient. So this is going from 15 down to 8. So that's a gap of 7. Whereas this one is going from 7 up to 5, which is a difference of 2. So this one on the left, the oxygen one, is like over 3 times the concentration gradient as the one on the right. That's what I mean. Cool. No worries. So many animals have specialized organs for gas exchange and transport. The diagram below shows the lungs of a mammal. Describe and explain how the lungs of a mammal are adapted for rapid gas exchange. Is that equation AQA? It used to be AQA, but it's no longer AQA. Although AQA do kind of expect you to know it. Weirdly, AQA don't explicitly tell you it, but you are expected to know that surface area increases the rate, concentration increases the rate, but diffusion distance decreases it. So AQA don't want you to know it, but they do want you to know it at the same time. Short diffusion distances, capillaries are 1 cell thick. Good blood supply, so concentration gradient is faster. Okay, good. Moist. Jess, tell me why moist is relevant. Large surface area. Cleo, you need to tell me why that's relevant. S, you need to tell me why the surface area to volume ratio is large. Is this OCRA? Yes, OCRA would expect you to know this. I wouldn't. I want to know more information about the alveoli being thin. I want to know why they're thin. Alveoli recoiling, yes, but you'd have to tell me a little bit more about why it can recoil. Surfactant, I would want a little bit more about surfactant. A good network of capillaries of short diffusion distance, that's good. Dissolved gases, capillary beds with capillaries being 1 cell thick, that's good. Rich blood supply, that's good. I want to know why a rich blood supply is useful though. Good, surfactant is now sort of been answered. Big cavity to allow lots of air to move in. Pleural cavity, yes. So hang on, I would go through this and sort of talk through, right? How does it have a large surface area to volume ratio? So if you get a question like this, you want to tick off surface area to volume. So how can I do that? Concentration gradient. And you want to tick off diffusion distance. If you do that, you'll get 4 marks. So loads of you have already started this. So if you say millions of alveoli, because there are millions, provide a large surface area to volume ratio, that'll get you 1 mark. So what most students do here is they'll say one or the other. So they'll say it's got a large surface area to volume ratio. That will never ever get you the mark. So you need to link up the structure, alveoli, to how that helps it to function, large surface area. How does it have, okay, I'll help you with the epithelium, endothelium. How does it have a high concentration gradient? How does it have a high concentration gradient? O2 is being used up in what? In fact, O2 isn't being used up. O2 is being something else happening. Good. Phoebe, you are right. So oxygen isn't being used like it would with an insect where it's used for respiration. Instead, oxygen is being moved into the blood, into the hemoglobin, and then it gets moved off. So continuous flow of blood maintains O2 and CO2 concentration gradient. What else helps with the concentration gradient? Yeah, good capillary network is good. Good removal of blood's fine. Is there a steep concentration gradient by having a higher atmospheric pressure outside of the alveoli than inside? No, because that would only ever force it in. Capillary, well ventilated. There you go. That's a good word. So ventilation, which just means breathing, which is why I always thought it should be the ventilation system as opposed to a respiratory system, but oh well. Ventilation, so the movement of gases in and out, that will also maintain the concentration gradient. Okay, how are we dealing with diffusion distance? Nothing to do with osmosis or anything just yet. Yep, I'll explain how moisture helps as well. Good, right. What is the outside of a capillary made of? Surface tension is something to do with surfactant. Yeah, I'll give you that. What's the outside of an alveoli called? What's the outside of a capillary called? What's the outer wall called? They are one cell thick. What's it made of? Can anyone tell me? Which one? Alveoli is epithelium. Good, right. So alveoli have one cell thick epithelial layer. And we want to say capillaries have one cell thick endothelial layer. So I don't know if there's an easy way to remember this, right? I've never heard a good way of remembering it unless someone knows it. But epithelium is sort of what every organ in your body has as the outer layer. So your gut has epithelial cells, like all of your organs have epithelial cells. The only thing that has the endothelial cells are your capillaries and blood vessels. So endothelium, you've got to somehow get it in your head that that's associated with blood vessels. Yeah, endothelium is for all blood vessels. So epithelium, epi, is in your organs and like various tissues. Endothelium is in your blood vessels. So you just want to work out a way that endothelium is your blood vessels. So I don't know, maybe like endo, like it gives me the impression of inside, you know, like endo and exocytosis. Endo is like inside. Maybe you can picture poking into your blood vessel or something. And I don't know, like did any kids in your class ever do that thing where like you were dissecting a heart and someone put the aorta around their finger and wore it like a ring or something? Kids used to do that. There you go. Someone's put their finger inside of like a blood vessel. I don't know, something like that. Endo blood vessel. Yeah, that's a good one. So endo has got an O. Blood vessel. It's got an O. Something like that's good. Someone said their teacher said something good. Hang on, I want to read that. Where was it? My teacher says the endothelium doesn't touch things from the outside as it's endo inside. Yeah, no, I see that. Teacher says endothelium doesn't. Yeah, that makes sense. What year is this paper? I have no idea. Right, anyway, you need to think of a way to remember this. So some other things that you could have said as well then. This was 2 marks, those bottom 2. So that is 5 marks there if you wanted it. So you could have said that there is warm air in the body. So you could say that by being internal, it warms the air up. And by being warm, you're going to help the rate of diffusion because there's more kinetic energy. So that's something. You could have said that the lungs are moist. And by basically being moist, you're preventing water loss. By being moist, you're basically doing what your xerophytes do, your plants, where they've got lots of water around them, which prevents water loss. You could say something like that. Aorta, that's how you pronounce it. You could talk about surfactant, which is going to lower the surface tension, which is going to help the lungs to function. You could say, I don't know. Oh yeah, I might actually be about it. Oh, you could say that there is elastic tissue. That's another good one, which I quite like. Elastic tissue in the alveoli is going to allow recoiling, which is useful in expiration. Cool. So yeah, other ones, warm air is going to speed up diffusion, or they're moist to reduce water loss, something like that. Do they have receptors? There'll be various proteins on the outside of them, yeah, but I don't know what's that's too relevant to. Yeah, so surfactant helps sort them sit together, surface tension, stuff like that. Right, this is relevant to some of you, but not all of you. So figure 1.2 represents the volume changes in the lung of a human. Select letters A to H, which correspond to the following diagram. First one has been done for you. AQA do not want you to know this. No. AQA, your teacher might have persuaded you that you need to know this, but you don't. With OCR, it's quite important. Yes. Yeah, OCR, love this. They talk about it a lot. So residual volume. So what does residual even mean? Like, you can normally work it out from that. EdXLA, yes, pretty sure they want you to know this. What does residual mean? It's what's left over. Good. So what's the one of these which is showing you what's left over after you've like had a big breath? Yeah. So look, H is showing you that after you've had the biggest breath that you can feasibly have, because that's how these movements are sort of caused, H is the residual bit that is left over. No, AQA not at all. So residual volume is H. Right? You might have heard this referred to sort of something to do with dead space. No, AQA don't want you to know this. Lots of AQA teachers still teach it for some unknown reason to me. I think lots of teachers forget to look at the spec sometimes. Total lung capacity. Which one shows all of the stuff that your lungs can maintain? So everything that your lung can hold. So the total lung capacity, literally total, all of it. It's got to be D. Right? That is the total of everything. I can see why you'd say C, but that's not all of it. It doesn't include this residual volume. So the total lung capacity has to be D. Tidal volume. Tidal volume is an easy one to remember. Yep. Tidal volume is F. It literally even looks like the sea. Look, you can see little waves. It literally looks like the sea. Right? Tidal volume is just the amount of air moving in and out for every breath. So F. And then vital capacity. Well done, Toby. So the vital capacity is the deepest breath that you can have, and then the biggest exhale, which can then precede that. So C is the vital capacity, is literally everything that you can feasibly breathe in and then subsequently breathe out, apart from this residual volume, which sort of we'll talk about in a second. So there you go, H, D, F, C in that order. Yeah, not a bad format. For OCR and for EdXLA, the ones that they care the most about are tidal volume, vital capacity, and residual. In fact, no, yeah, residual volume. They're the ones which come up most likely. It's mostly these bottom two that they care about: tidal volume, vital capacity. Residual volume is easy to remember. Total lung capacity should be something not too bad to remember. Okay, figure 3.1 shows a trace recorded from this apparatus. Calculate the rate of breathing over the first minute from this trace. Trippy graph. It is making my eyes go a bit weird, yeah. I used to have loads of kids who used to say, oh, I can't actually read a graph because I can't see the little squares. I always used to think they were sort of moaning, but that does look quite weird to me, to be fair. Okay, so the minute mark comes up to here. So how many peaks can you see? One, two, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13. So 13 whole ones. In fact, we're sort of going from a trough, aren't we? So if we're going to this bottom trough, there's my first trough that I've gone from. Now, obsessingly for all of you guys, we've got to include that bit as well. Doesn't actually matter what you use. So if I'm including this bit, obsessingly for you guys, it's actually 13.5. But how do you do it? So I would look at what you are on for 60. So for 60, I'm looking at a trough, right? So I'd probably start at the first trough. So the first trough is here. And then there's sort of half of a trough here, because there would be one just about here, which I'm just about missing. So it's half. So 13.5. So the mark scheme literally says 13.5. You can't get 13, you can't get 14. It's 13.5. If we're doing the rate of breathing, we would be doing breaths, and then maybe minute -1 or something if you wanted to. Or you could write it out in full. Breaths, minute -1, something like that. That's really how I'd write it down as a unit. But yeah, 13.5. The mark scheme literally just doesn't credit anything about that. So be careful. Okay, so about 1 decimeter of air cannot be expelled from the lungs. This is known as your residual volume. Suggest why it's not possible to expel all of the air from your lungs. So if I got you guys to breathe out as deeply as possible, why can you not get rid of all of the air in your body? So surfactant does help to prevent the alveoli from collapsing, that's true, to prevent them from literally sticking together. So they would collapse. Why is it physically not possible? You're telling me why you shouldn't do it. Why is it not possible? Why is it not physically possible? So I like what you're saying, Phoebe. That is a good answer. You don't have quite enough force. You don't have enough force to do what? The air pressure outside doesn't allow it. No, because you can make the internal air quite large. Why can't you completely... Why can't you make that really high pressure? Yeah, why can't you collapse your airways entirely? In fact, you're actually not far off for saying that. Tell me some of the things that's like, so if you were to fill your... I don't want to say anything. No. Good. Jess Smith, I'll give you that. So one thing that you could say... Don't know why I'm writing a J. Your ribs cannot completely compress. Right? If your ribs could smush together entirely, then you would be able to get rid of all the air. That is one thing which is going to prevent you from expelling all of the air. What do you know about your airways? I don't want to use any words. If you feel your neck, what can you feel? What about your C-shaped cartilage? What about it? It's solid, therefore, yeah. Like, can you feasibly do it? I can't. If I like do this and focus, I can't cause my trachea to completely flatten. So even if I could completely flatten my lungs, which I can't, my cartilage is always going to be open, right? It's always held open. So it's going to prevent them from collapsing all the way. So your ribs cannot completely compress. Your cartilage... Not up. Holds open your trachea. And that's, to be honest, that's like the main points. I think feasibly you could get one for saying that your alveoli can't compress all the way as well. But I don't know what you'd want to say. Maybe surfactant. Yeah, go on. Let's talk about surfactant as well. Surfactants prevents alveoli collapsing too, or something, or sticking together. Right. If you haven't come across the word surfactant in your life, then pretend I've never said it. Some specs use it, some don't. What is surfactant? It's like this fluid stuff, which I think is made of like some form of lipid and some other bits. And it basically stops it all from sticking together. So your alveoli from basically coming together. It basically removes surface tension. Your C-shaped cartilage holds open trachea, your ribs can't be compressed. Yep, nice, that looks good. Vidori. Well, for this question, surfactant's fine, but pulmonary surfactant is a better way of describing it, so yeah, why not? Okay. I used to love this model. So many teachers use models to demonstrate and explain the breathing and lung function in mammals. Figure 2.1 is a model of a mammalian chest. When the rubber sheet is pulled down, the balloons expand. Explain why the balloons expand. So if you pull down on this little, have you guys seen this before? It's like called a bell jar. You have one teacher hold it, like it's really heavy in one arm and you sort of pull it with the other one. So why are the why are the balloons gonna expand when you pull it? Huh. I do use physics and maths tutor every now and then to get some questions. Right, so the pressure outside the lungs is decreased. How? The pressure outside of the lungs. So you're saying, right, you're saying the pressure in this region, right? Don't say outside of lungs, try and be specific. So the pressure in the jar, right? So increased volume, so low pressure. Very, very good, Talia. Internal pressure is greater than outside. Very good, Phoebe, for mentioning that pressure is like, you don't just say one. So go on. Essentially what's happening then is they are going to pull this plastic sheet down. So it's not solid. It's like, well, it is, but there's a bit of plastic which can be sort of moved. So you're going to pull it down. When you do that, we're going to say that the volume of the jar will increase. Okay, now, I don't know if you guys will know the guy who came up with the law, but Boyle's law states that volume is inversely proportional to pressure. So if I increase the volume, then the pressure inside the jar decreases. And then if we're explaining what's actually going on now, we'd want to say, you'd want to essentially say that the pressure in here is now less than the pressure out here. So whenever you talk about pressure, it basically doesn't make sense unless you compare two places. So you want to say that the volume has increased, the pressure inside has decreased, and then it's really, really important that you say that external pressure is greater than internal pressure, which is going to cause air to be forced in. Right? So whenever you talk about pressure in biology, literally any time you want to use the word pressure, so whether it's transpiration or whether it's translocation or whether it's the heart or whether it's this, right? You always need to talk about pressure in two different places. So the external pressure is greater than the internal pressure, forcing air in. So yeah, you've made a gradient. This is, you could say here that there is a gradient established between external and internal. Pleural membranes are going to contract, not relevant here. Pressure gradient is created, air is pulled in. Yeah, these balloons. Yeah, these are literally just two balloons. Before this happens, the pressure in the balloons is equal to the jar. Yeah, before this happens, it's not really the pressure of the balloons that I care about, right? If it's at equilibrium, the pressure here is going to be the same as out here. But what I'm trying to do is I'm trying to make the pressure here, so in the thoracic cavity, or what would be the bell jar, I'm going to try and make this less than this. Okay, so I've got a great pressure outside, got a lower pressure inside. Pressure's it's going to cause it to move in. That's why you have wind, in case anyone's never explained to you why wind exists. You have high pressure over here, low pressure over here, so wind goes that way. One of those things that you never really think about. Right, anyway, easy question, massive amount of space for some reason. Describe a pathway taken by an oxygen molecule from the alveolus to the blood. So what is the pathway that oxygen takes from the alveolus to the blood? See if you guys have learned those things we said earlier. Night hardcore, I like that you, I'm glad you like that fact. Do you know why you get waves? Do you know the main reason why you get waves? Most people don't know this either. O2 diffuses from the alveoli into erythrocytes and binds to hemoglobin. Namrata, you'll get no marks for that. Diffuses through the capillary. It does, but I want more. Good. I don't know how to pronounce your name. Is it Freya or Freya? I assume it's one of them. No marks for you, Vidori. Freya. Okay, cool. I assumed it was Freya. I'll remember that. So describe a pathway. You want to say that O2 is basically going to move from the alveoli, alveolus. O2 will move through alveoli. I don't know why I've done it as capital. Epithelial cells. And through capillaries, endothelial cells. Into an erythrocyte or into a red blood cell or something. Yeah, that's how it works. So to get your 2 marks, they wanted epithelium and they wanted endothelium of capillary. So do make sure that you know the difference between endothelium and epithelium. Do you need to say down a concentration gradient? It is technically down a concentration gradient. It's not in here, but why not? Why not add it? If you know another fact, why not add it in, right? Wind question. The wind question. Reason you get waves on the sea is because of wind. Okay. So you know, like if you've ever seen a sand dune and you've seen sand and it forms these weird patterns because of the wind. Same reason why you get waves. Anyway, stomata are found mainly on the underside of leaves of dicotyledonous plants. A student painted a layer of nail varnish on the lower epidermis of a leaf. She peeled off a dry layer of nail varnish and placed it on a microscope slide. The student was able to see the impressions of the stomata on the varnish using an optical microscope. She then determined the mean diameter of the stomata. Describe how the student could use an eyepiece graticule to determine the mean diameter of the stomata. How can you use an eyepiece graticule to work out the size of something? Common question people hate. So yeah, maybe some of you might not have come across this before. So an eyepiece graticule, so you get little microscope slides which have little things on them, which I'm not going to tell you just yet. And eyepiece graticules, if you've ever looked down a microscope and you've seen a line with little numbers on it, that's an eyepiece graticule. This is an AQA, yeah. Line up the graticule and the micrometer and work out the difference between divisions. I don't actually know if it is an OCR. I think it might actually be one of the sole things, which is like AQA only. Is not Oli. Okay, cool. It is OCR. Okay, some of you may have done this. Right, to get away with this question, right, you get a really nice, easy little mark. So this doesn't want you to tell me how to really do this. All you need to know is that the word we use is calibrate eyepiece graticule to a stage micrometer. There you go, you don't need to tell me any more than that. Right, in real life, how you do it, if I know that this distance is exactly 10 micrometers, right, and I've got an eyepiece graticule which has, I don't know, that many units. There you go, that rough, in fact. There you go. So I know that it goes 1, 2, 3, 4, 5, 6, 7. So 10 micrometers is about 7 of those little units. So I know that one of those little units is going to be 10 over 7. Whatever that is. Right, so that's how you know how big it is. So you calibrate your eyepiece graticule to a stage micrometer. What would you do next? Eyepiece graticule. There you go, that word there that I highlighted. Oh no, this is definitely AQA. Yeah, 100%. So once I know in my little microscope, so when you look down your microscope, here is your sort of thing that you look down, your eye goes somewhere here, right? There's your eye. Right, you look down your microscope. If I know that each of these little things here, let's actually give it a proper number as opposed to 10 over 7, because no one likes numbers like that. So if I know that each one of these is 1.4 micrometers, so each of these is 1.4 micrometers, and I've got some stomata like all over the place, how can I work out how big a stomata is? Yeah, so see how many. Well, I realise I've got a load of questions left. It's almost 4. Jesus. So see how many stomata fit. Stomata. In the eyepiece graticule or something. So I don't know. That's actually a stupid way of saying it. Let's say measure the stomata against the eyepiece graticule. Would I only want to do one? Would I only want to do one? Good. Yep. Find a mean. So multiple times, repeat. Repeat multiple times, find mean. Okay, and then you, I don't know, in fact, that's about it really. Then you would multiply your number of eyepiece graticle units by 1.4. So I'm going to leave that there, but you can get the format of saying that. So calibrate your eyepiece graticule to a stage micrometer, measure the stomata against the eyepiece graticule, repeat multiple times, find the mean, and then you can use that to find the size. Right, I'm going to do one more question. I'm not going to do the other ones, so you guys can do the other ones another time. In fact, I'll tell you the answer very quickly. So this one, forced expiration, all you need to say was contraction of internal intercostals, relaxation of the diaphragm, decrease in volume of the chest, air is pushed out due to the pressure gradient. This one, appropriate units. If you were looking at the distribution of stomata on leaves, that one is just stomata per millimeter or something. One last question, just to check if you guys have done this. So one of the 2 plant species used by the student in an investigation was a xerophyte. Other than the distribution of stomata, suggest and explain 2 xerophytic features that leaves have. Yeah, sorry to rush through those last ones. So what things does stomata have? Go on, see what you guys know. Yeah, Monday and Wednesday. Thank you, Karen. Oh, guys, you know this. So what's the point of these things? Before you think we're done, what's the point of these things that you're talking about? So what's the point of hairs? What's the point of a thick cuticle? They prevent water loss. How? Good. Trap water vapor. Reducing water concentration gradients. Preventing water loss. How does a thick waxy cuticle reduce water loss? Good, yeah, it blocks transpiration. So it prevents evaporation of water. Very well done. There you go. Ultimately, when it comes down to xerophytes, they either do 2 things. They either trap water, and then you can just say the same sentence again, reducing water concentration gradient, preventing water loss, or they prevent evaporation. So they either do one of those things. So stomata in little pits reduce water loss because it traps water and it prevents there being a concentration gradient. Folded leaves traps water vapor, reduces water concentration gradient, prevents water loss. Having fewer stomata prevents evaporation of water, right? All of them are the same, right? Literally always the same. Right, guys, I am going to tell you something really exciting. So if you've ever felt like the thoracic cavity, which normally I do, normally I'll get home from a day of work and I'll feel like a thoracic cavity because throughout the day, the volume has been changing. The volume of things that I need to know is changing. It's getting ever greater. But unlike Boyle's law, that would imply that the pressure on me would be getting reduced. However, for me, it feels like the pressure is building up. So the volume's increasing of knowledge, but the pressure is also building. Right? There's more pressure for you to know things. Wow. Luckily for you, almost like, let's go for forced inhalation. Almost like forced inhalation, SNAP revises here to help you to revise. It will force information into you, to your brain, and that will help to relieve the pressure on you because I didn't think this far ahead. I thought, I know, I can work out a link here between volume and pressure, but I didn't think it through. No, I'll work it out. Snap revise will be like inspiration because it will force air in, which implies that the pressure outside is less than the pressure inside. So internally, it reduces the pressure on you. Despite the pressure being hard outside to get into university and to do well in school, the pressure inside will be nice and calm. There you go. Knew I'd be able to do that. So for those of you that are not familiar with Snap Revise, I have some information for you. Basically, not only do I do these web classes every Monday and Wednesday, and not only does Georgia do chemistry on Tuesday and Thursday, and not only does Max do web classes in maths on Sundays, which I can't remember, and not only does Matt do physics, but we also do a whole lot more, basically. So let's pick randomly OCR, A-level biology, right? For instance, today tomorrow that is, I'm doing a lesson on fractionation, mitotic index, and drawing biological specimens. The same day at 3 o'clock, I'm doing permeability practicals. On Wednesday, I'm doing statistical tests. On Thursday, I'm doing more membrane stuff. On Monday, I'm doing circulation, clotting, cardiovascular disease in the liver. Basically, every day I do at least one web class. In fact, every day I do at least 2 web classes, and some days I do 3. So I do drop-in sessions too, where I don't know. Look, I'll show you the one I did today. So someone wanted to go over hemoglobin and gas exchange, so I spent my day going over hemoglobin and gas exchange for about an hour. Right? I do a book club. I've been going over AQA essays because I know lots of people don't like AQA essays. Been going over literally whatever people want. I just go over it. I do the same with like exam questions too, where a kid will say, "Oh, can I have an exam question on this?" And I'll say, "Yes, of course." So that's what Snap Revise does. However, that is literally the very tip of the iceberg, as I'm sure you know. A few other things that we have are we basically have a system to work out what you do and don't know. So say you're one of these people who, I don't know, you're sat in year 12 and you're thinking, I think I know this stuff. And then you go and do an exam and you realize, oh God, I thought I knew it, but clearly I knew nothing. What went wrong? Well, this is where like part of our other packages come in, right? So we have lots of quizzes. So from what I've learned about educational theory, essentially, if you don't try to recall information, you are basically as good as not having done very much at all. So in revision, lots of people just write things down or they just read things, and that is dreadful revision. Essentially, what you need to do is you need to have little means of assessing yourself. So we have little means of assessing yourself to see what you know at the very start of each topic. I'm not going to go through it, but you have a little diagnostic test, right? After you've done that, you get a little thing pop up in yellow if you haven't understood something. So where I didn't actually do that full quiz, I have nothing coming up. But for instance, it will say that I don't know much about the formation of proteins. So here is some bloke talking about proteins for a little while for like 10 minutes, 8 minutes. After you've done that, you can do another quiz to check that you've actually learnt anything. And if you haven't, you can go back or you can complain or something. It then goes on through the next things, through the next things, through the next things. You get a quiz, and then finally you end up having an exam question. So someone will go through an exam question with you on whatever it is. And this covers the entire specification that you have. So AQA, Edexcel, A, B, OCR. When you're done with that, say you thought, oh God, like I'm still not entirely convinced. We have exam packs. So here are a whole load of questions about proteins that you could have a look at. And then here is an answer. And then it's the same for literally all of them. So there are some answers. Let's say that even after that, you're still unsure. You could go back to a video and you could, I want to find one where I've actually answered a question. Basically, if people don't get things, there you go. Someone said that the screen went black for them. They can write a comment and I can get back to them and tell them how to fix it, right? I don't know why this was happening. Apparently, it wasn't happening for me or any of my colleagues, but I think we fixed it. So if you have a problem, you can basically write on here and I'll get back to you within about 24 hours, less of a weekend, in which case I'm chilling. Finally, there is a revision guide as well. So if that wasn't enough, which I'm sure it is, you can also have a free revision guide thrown in too. So for those of you that are at all interested in that, then here is a code for you so you can get some money off. Essentially, we have different packages going from the cheapest one being about £10 a week sorry, a month, and the most expensive one being about £50 a month, which I know is a lot of money, and I don't want to sort of catch you off guard, but you're getting at least 48. I think I worked out at least 48 web classes a week that you could turn up to. And as someone who used to be a private tutor before I was a teacher, I used to charge about 40 quid a lesson. So it's actually pretty good value for money. I know it's a lot nonetheless. And if I haven't convinced you and you don't want this, and I'll come back to this in a second, I'll answer your questions, then at the very least, it would be lovely if you guys could turn up to any more of our YouTube sessions. So hang on, if I just load this up, I'll show you where we're at. And don't judge, this is the office's YouTube channel, and some people look up some weird things like pitching for Dragon's Den. But yeah, here's what's coming up, or in fact, here is what's coming up in the next few days. So for instance, we have physics, which is at 12 today. I was at 12 today. This one, oh no, that's not right. What's going on? 5:12. Is this the American way? Yeah, that's tomorrow. So that's the 12th. That is also the 12th. That's the 13th. That's the 13th. So look, we've got loads of things coming up. So I don't know. If you do biology, maybe turn up to hemoglobin. If you do chemistry, maybe you want to go over the ideal gas and other calculations, equations. Maybe if you do physics, you'll catch up with, I don't know, some moments. So if I whack that back there, feel free to use this. It does expire tonight, but feel free to use it. Otherwise, I will just sit around here and answer any questions. What are we saying? My laptop is powerful enough to stream videos about lag. My laptop is, I don't think my laptop matters. I think it's my internet, no? I should wear caps more often. You look cool. Someone told me that earlier today. Thank you, Massimi. That is very kind of you. You would be impressed. Maybe not impressed. That's the wrong word. I wore a bandana for the first time the other day. I went for a run and I bought a little bandana, which looks like Rambo. So I went out like that. And then I thought I'd wear it like with my friends on a group catch up. And they ripped into me the whole time. So unfortunately, I'm going to have to make sure I do that again just to get them used to it. What else saying? Dragon's Den, yes. Do you save the live streams if we can't go live? On the website, yes. So hang on. I'll show you quickly. So on here, right, let's say that you wanted to look at some videos. Hang on, where am I at? Here I am. I definitely know how to navigate this properly. So here are all the classes that I've done recently, right? But here are all the classes that I've ever done. So if you wanted to learn a bit about, I don't know, codominance, multiple alleles, and sex linkage, all you need to do is click on that. Then here is a video of me talking about those things for however long you want. There it is. There's me without a hat, with shorter hair, back when I could cut my hair whenever I wanted to. Lovely. If you were mentioning the ones on YouTube, then equally, yes. There are actually some which are a lot older than you guys would probably realize. So we have a lot of videos, obviously, but I did some like months and months and months ago. Like I did one on I did one on carbohydrates, like at the very, very start of this year when I started working here. And we do obviously post quite a lot of things, but if you go back far enough, you'll be able to find some of the earlier ones. So this one's got 3.8 thousand views just on enzymes. Proteins there, carbohydrates, lipids, 3.5 for carbohydrates. Yeah, so going back to 7 months ago, we've got videos that you guys can watch, right? The more recent ones, like the kidney, there's one from 3 months ago, you can watch that. They're all here. Like you don't have to search too hard to find any of them, I don't think. What else then? In anaphase 1, do homologs separate and then move towards poles? In anaphase 1, do you mean meiosis or mitosis? Unknown. Can I see you on the web classes? I cannot see you on the web classes, no. So I was having a go the other day at listening. So I do a book club once a week, and we're reading The Immortal Life of Henrietta Lacks, who's this really interesting woman from the 50 or 1940s, 50s-ish. And I had a go the other day at letting the other people talk. So I normally have about, 5 to 10 people in my book club sessions. And yeah, I let them talk the other day, and that was really cool. So I've never obviously heard any of my students' voices before. But no, I cannot see you. And we thought that would probably be easier to stop people getting distracted. And also to make sure that you don't have to do your hair or, make sure you're wearing something nice we wanted to get rid of that. So no, we can't see you. It's just me. It's like this, but it's just on Zoom. Unknown, no worries. So what do you have to do for a free account? I'm pretty sure you just need to go onto a website and it will give you a free account for a bit. Meiosis. So yeah, essentially what happens then is you have your, what was the previous question? Do homologs separate and move towards a pulse? So anaphase 1, yeah. So you should know then that in meiosis, all your homologous chromosomes, so I wouldn't say homologs, I'd say homologous chromosomes, they've all lined up together, they've all done their crossing over, and they've all done the whole producing new allelic combinations. And then in anaphase 1, your homologous chromosomes move towards the poles because they're being pulled by spindle fibers. So they're pulled at the centromere and they're pulled towards the 2 poles. And then in meiosis 2, you end up with your chromatids being separated, which are also called part of their centromere. Thank you for saying my hair looks cool. What else? Could you give me advice? I'm in year 11 starting biology now. Ooh, yeah, advice. Make sure you know year 11 stuff very well. So I think the worst thing that you could do as a year 11 student is start trying to learn A-level biology before you've properly learned your GCSE. So I imagine lots of people are going to be in the same position as you, where they think that they're better off learning learning A-level and getting ahead. But if you haven't got the foundation GCSE, you're going to struggle. So make sure you know mitosis, make sure you know all your cell stuff, make sure you know your genetics, make sure you know pretty much all of your GCSE so well, pretty much as if you'd had an exam in it, that when you move on to A-level and start looking at that, it's not ridiculously hard, because the jump for GCSE to A-level is already really big, and that's implying that you know all of GCSE. So on average, if you got an A at GCSE, then you're probably going to get a B at A-level, right? If you got an A star at GCSE, you're probably going to get an A. Obviously, there's some exceptions to this, but that tends to be what we as teachers tend to see. So I would say make sure that if you say you want to go into medicine or something where you're going to need at least an A, but actually probably an A star, you need to firstly make sure that you're already an A star student at GCSE. And if you're not, you're going to struggle. It says sufficient data for now, but it's just waste of time. I'm very confused. Okay, what were we saying? So wait a second. No, we need English Study Club. Yeah, English Study Club would be good. I don't think we have that yet. Yeah, I've started struggling quite a bit. I've done my biological molecules. Yeah, okay, yeah, biological molecules starting off with carbohydrates. There's not too much. I think it's you've got to start getting used to words that is less important at GCSE. So a big step with GCSE going up to A-level is that at GCSE, the words you use don't matter so long as you're descriptive. At A-level, if you don't use words like hydrolysis, if you don't use words like condensation, you're going to lose all of your marks. So key words are very, very, very important at A-level. You got a D at your GCSE. Ah, okay. Well, hopefully you're working really hard now, me maker. Is there any oxygen in deoxygenated blood? I've read that it stays about 75% saturated. Yeah, there'll be a little bit of oxygen in deoxygenated blood. Okay, yeah, no, HA, just make sure you really know your year 11 stuff, like as well as you can. You got 8.7 at GCSE, but I average like D or C. Yeah, no, that's common. I think most people in year 12 do quite badly. So back when I was at school, back in the day, I sat my A levels in 2012. You could reset. So at the end of year 12, I think I had a D in biology or a C in biology. I really wasn't doing very well. I wasn't trying very hard, I don't think, or I wasn't trying hard enough, clearly. But I ended up getting an A. So I was lucky that I could reset and make sure that I brought all my year 12 marks up. And now, obviously, you guys have all of your exams in year 13. So I was fortunate in that sense. But yeah, most people finish year 12 not as good as they finish year 13. You can normally get at least a grade up. I think I smashed it just because I had a goal, like I really wanted to get to my university. That made life easier. Best way to revise immunity. I've done flashcards and practice questions, but I'm still struggling. When you say flashcards, there is a way to make flashcards. There's a right way and there's a wrong way. So flashcards should be one topic. That is well concise. That's definitely not well concise. Whatever. Basically, I'm going to get rid of that before I look more of an idiot. You need to have one topic, right? So one very, very brief topic, which you have summarized as much as you feasibly can. So that's one side of your flashcard. The other side is a question, right? One exam question on the other side. And basically, if you cover the entire topic with exam questions on one side and like a really simplified version of it on the other, like phagocytosis or B cells or T cells or whatever, then essentially what you do is you read it, you do the question. If you can't do it and you don't get it right, then put it at the back of the pile and go through them again and keep going through them until you know them. So I'd say with immunity, you just need to assess yourself a lot. Feasibly, my favorite word. Oh God, I've become, is it C? It was C. I've become one of those teachers. I used to have a teacher who used to always say the same thing. She used to always say, you need to know constantly. Like, you need to know, you need to know, you need to know. And I always like, oh no, I'm never going to be that person who just repeats myself all the time. But apparently I feasibly have become that person. Did you practice more exam questions or content revision? I did both. So I used to save up exam questions as a little treat at the end of a week because they were the easy bit, right? It was a revision, which was hard. The other things are hard. Can you watch free web class with a free trial? I don't know, Annabelle. I'm not entirely sure. You might have to check it out. Right, anyway, I'm going to head off now, because otherwise I'm going to sit here all evening. So have a lovely afternoon, guys. Any more questions you got, just let me know and I'll answer them next time. Yeah, I'll catch you on Wednesday. So have a really nice rest of your day, and I will see you later. Bye.